Hi everyone, I'm Dr. Taseen. I'm sure you're all wondering why it's taking so long to develop a vaccine against COVID-19. When we talk about vaccines, it's hard not to mention Dr. Edward Jenner. He was one of the pioneers who really established the concept of vaccines when he vaccinated this 8-year-old boy in about 1796. And he demonstrated that using vaccination you can protect against infection. There are several types of vaccines. I think it's really important to talk about the types of vaccines. This includes live attenuated vaccines, inactivated or killed vaccines, subunit vaccines, toxoid vaccines, as well as DNA vaccines and recombinant vaccines. Live attenuated vaccines are essentially a live pathogen or a live strain which has been modified so it's less virulent or less dangerous. Some examples include the famous MMR vaccine, which is the measles, mumps, and rubella vaccine. You also have the rotavirus vaccine, the smallpox, chickenpox, and yellow fever vaccine. All these vaccines are live attenuated vaccines. The second type of vaccines include inactivated or killed vaccines. This is essentially a vaccine which consists of a killed strain of the pathogen so that you don't have any there's no live organism within that vaccine, so a lot of people feel that they're safer. Some examples include hepatitis A vaccine, the flu shot, the polio shot, or the rabies vaccine. Third class of vaccines include, these are basically subunit vaccines or components of pathogens, components that include surface structures of pathogens. They are usually very well defined vaccines. This is when you know exactly what is required for protection against a pathogen. They're quite successful. They include the Haemophilus influenza vaccine, the hepatitis B vaccine, the vaccine for Bordetella pertussis protection, which includes protection against the whooping cough. Some of these vaccines can also be conjugate vaccines where the protective component of the vaccine is conjugated to some other protein. They also include vaccines which include components of polysaccharides or capsules, which are the surface structures of some pathogens. The fourth class of vaccines includes toxoid vaccines. These are very famous and effective vaccines. They include components of toxins which are inactivated. They're quite effective. They're effective against preventing the effects of the toxin. However, they don't always prevent transmission of the pathogen. Some examples include the diphtheria vaccine and the tetanus vaccine. The last class of vaccines I want to talk about are the DNA and recombinant vaccines or recombinant vector vaccines. These vaccines are thought of to be some of the feature vaccines. A lot of work is being done into DNA vaccines. DNA vaccines are essentially DNA that is expressed on a plasmid and delivered to a certain area of the body where the plasmid is expressed, and they hope that you get protection against the pathogen of interest. Recombinant vector vaccines are essentially technologies that allow you to express your antigen or your protein of interest, and it's delivered to a certain area of the body, and then you look for protection against the pathogen. What are some of the factors that you need to consider before developing or producing a vaccine? This is really important because you want to be able to design a vaccine that's protective and safe for use. Some of those factors include the type of immune response needed, animal models that are available, the target population. This would guide your approach to developing a vaccine. And finally, you want your vaccine to be safe so that it does not have any side effects and can be used effectively. When you look at the type of immune response, this is really important because when you look at the immune response, there are essentially two components to it. You want to find out if you need an antibody-mediated protection, or you want an antibody-mediated response, or do you want a cell-mediated response. And this would be studied, would be figured out using, several approaches. Ultimately, you want a strategy that is going to clear the infection. So based on that, you want to, and in some cases, you need a balanced response. You want an antibody-mediated as well as a cell-mediated response. Animal models are extremely important in the design or the production of vaccines. They allow researchers and microbiologists around the world to really study the pathogenesis of the organism and devise a strategy which is going to inhibit or limit the infection. Depending on your target, you may use mouse models to develop vaccines, which have been used for a long time. Some people use the porcine model, the bovine model, chicken model, and some people even use monkeys. The target population is extremely important because if it's an animal vaccine, you have to follow certain protocols for getting it licensed. If it's a human vaccine, it's definitely more difficult. You need to follow certain guidelines and regulations, and you need to prove that the vaccine is effective, it's safe. The number of immunizations is important. None of us like to get vaccines, so if you can get everything done within one shot, that is ideal, if not 2 shots. If you need more than that, it becomes expensive and difficult to administer. The age group is also very important. Are we targeting the elderly? Are we targeting the infants? Are we targeting age group that is in between the infants and the elderly? With all this information, you get to develop your strategy for making a vaccine, and this includes using the right technology so that you can produce large amounts of the antigen or the protein of interest. Do you need an adjuvant? Adjuvant are chemicals that enhance the immune response. If your antigen is giving, or your protein of interest is giving you antibody-mediated response, but you want a cell-mediated response, there are adjuvants, chemicals that can be added to your formulation so that you can shift the immune response. If you want a balanced immune response, there are also chemicals that can be used, adjuvants that can be used to do that. The stability of the antigen is really important. A lot of the times vaccines are produced in one part of the world and they're transferred or delivered to another part of the world. So the vaccine formulation has to be very stable so that it's effective and protective in the other part of the world. Temperature stability is also important. For some vaccines, they need to be refrigerated. So if they're going to be transported to countries where that is not always possible, those vaccines are not going to be effective. Vaccine safety is an extremely important area. Before any vaccine is permitted to be used or licensed, it has to go through several safety regulations. One of the first things is that the vaccine shouldn't be toxic. So when you immunize a person or an animal, there should be no real toxic effects. That person shouldn't become even, shouldn't become really ill or sick, should not display some of the classical symptoms of those pathogens. You might get a fever and a little bit, but not really the classical symptoms of that pathogen. You don't want really any side effects, some allergic reaction happening, some big blisters forming or something, not really unusual. You want to do many field trials. So once a vaccine is produced in the lab, it has to be, in the case of animal vaccines, you have to do several field trials. In the case of human vaccines, you want to do many human trials and get as much data as possible to satisfy the regulatory requirements and to ensure that the vaccine is safe. So with the COVID-19 vaccine approach, a lot of work is currently being done, and most of the studies, to my knowledge, are being done where they're using the surface or the spike protein, the S protein, which has been shown to be quite a good target for other similar coronaviruses like the SARS and the MERS. So I think with this approach, once they use that, it's still some time before we get the right solution or the right answer, but I think they are going in the right direction. Very soon we'll be hearing some promising data or promising results. Thank you so much for watching, and stay tuned for more videos.